I'll be discussing allergy, which is covered in chapter 14 in the book. And I'll be referring again to some figures in the book, and since the screen might be a little small, you'll see in larger font the figure numbers, so you can refer to that in your textbook for more of the details. So today, we're first going to be talking about, just in general, what is allergy? And what are different types of allergy? And some of these types we'll be covering include IgE and IgE-mediated allergic diseases. We'll also be talking a lot about what causes allergy. For example, what are some of the genetic factors that predispose someone to allergy? And also, what factors in the environment contribute to the development of allergies? We'll also be discussing some important cell types that you guys heard mentioned about at the very beginning of the lectures. But these cell types in particular are very important for an allergic response, so we'll be talking about that in detail. We'll also be talking about the difference between an acute allergic response and a chronic allergic response, and the differences in the immune mechanisms behind those different responses. And finally, we'll be talking about some non-IgE mediated allergic responses. So to start out, what is allergy? In general, allergy is considered a harmful immune-mediated hypersensitivity to a harmless antigen. And here, the key word is really harmless. So we've talked a lot through these lectures about how the immune system mounts a response to pathogens or to things that might damage the body. However, allergy is when these responses are directed towards something that otherwise wouldn't be causing any problems. So there are a couple different types of allergy. First, we have type 1, which is your immediate reactions mediated by IgE and by mast cells. And these are really the stereotypical allergic responses that you guys are probably familiar with, and we'll be focusing mainly on these today. We'll also talk about type 2 and type 3 allergy, and these are not mediated by IgE. They're actually mediated by other antibodies, primarily IgG, and they also can involve complement or effector functions after those antibodies bind to their Fc receptors. Then we have type 4 allergy, and this is driven by cellular effectors. So we'll be touching on this as well towards the end of the lecture, but this is really going to be focusing on cell types such as CD8 cytotoxic T cells. And really, these types of allergy, this type 1 through 4, is becoming a little outdated in terminology. This is what the book really talks about most, but nowadays people are moving towards more of a classification such as type 1, 2, and 3 immune responses. So typically you can think of allergy in the stereotypical way we talk about it as sensitization to a harmless antigen by the development of IgE. And this is usually mediated through a type 2 immune response. So if you guys refer, this is figure 14.1. This is a nice chart summarizing some of the IgE-mediated immune reactions. And here, some of these that you're probably already familiar with that we'll talk about in more detail in the lecture are systemic anaphylaxis, acute urticaria, or wheal and flare. This is also known as hives. Then we'll also talk a little bit about things like hay fever and asthma, which also has an allergic component. And finally, we'll kind of wrap things up talking about food allergy, as some of those responses are mediated a bit differently. Okay, so we're going to start out focusing on IgE and IgE-mediated allergic responses. So immediate allergic reactions, such as anaphylaxis, are caused by IgE binding to the Fc epsilon receptor, mostly on the surface of mast cells and basophils. And the Fc epsilon receptor is really unique amongst Fc receptors, and that's because it binds with very high affinity to IgE. And such high affinity that IgE is actually typically already bound to the Fc receptor in tissues rather than circulating in the blood. So when these IgE antibodies bound to the cell surface come into contact with their antigen, this antigen will cross-link multiple IgEs on the Fc receptors, and this will lead to degranulation by mast cells, which this is where mast cells release pre-made chemicals held in granules, and we'll talk about what makes up these granules in a bit more detail later. So here you can see this is just a nice summary of how this response works. So you see in the top part, the IgE secreted by plasma cells, and it binds to the high affinity Fc epsilon receptor on the cell surface of mast cells and basophils. Then once these cells are activated, they can also provide signals to B cells, and this works as a feedback mechanism whereby, So this acts as a feedback mechanism whereby these B cells then begin to produce more IgE. Okay, so I mentioned earlier the word sensitization to an antigen. So what does that mean? In particular, what does it mean for a response to be sensitized through IgE? So switching to IgE via class switching in B cells can first occur once an antigen is initially seen. So the first time that the body sees an antigen, typically allergic symptoms actually will not occur. It's upon subsequent exposures that the sensitization and symptoms will actually begin to manifest. So this is actually called Atopic March, and what that means is that typically individuals have a progression of allergic symptoms. So these symptoms can start out minor, maybe with some itching, but then they'll progressively increase in severity. And this can then include things such as hives, swelling, and potentially anaphylaxis as well. So not all allergen exposure leads to sensitization, however. So every person who comes into contact with an allergen, for example, peanuts, does not become sensitized and mount an allergic response to this. And also, not all sensitization leads to a systemic symptomatic response. So it's possible that individuals can have IgE produced to an antigen, but they will not have symptoms when in contact with that allergen. And IgE production is really occurring in response to type 2 signals. So a type 2 immune response will skew B cell class switching towards an IgE response. And I'll be using the word atopy as we go throughout this talk. And atopy just means that someone is predisposed to IgE sensitization. So we can see here, allergens, I mentioned before, are typically harmless things that the body comes into contact with. So some features of these airborne allergens actually help skew towards a type 2 response, leading to allergy. So for example, proteins with carbohydrate side chains might induce a T cell response, which then can provide help to B cells. And also, if these antigens are encountered in a low dose, this will favor activation of IL-4 producing cells. It's also possible that these antigens might be highly soluble, which means that even though they might be bound to a particle, such as a granule of pollen, they're very easily removed from that granule into the body. And often these antigens are also very stable, so they can survive being on that pollen granule for long periods of time. So these are just a few features that are often characteristic of airborne allergens in particular. So very quickly before we move on, I've mentioned type 2 response. So I just wanted to give you guys a quick overview, a review of type 2 immunity. So here we can see this figure is from a paper in Science Immunology that we have our eosinophils, which are very important for allergy, as we'll talk about. And these eosinophils are produced primarily in response to cytokines such as IL-4, IL-5, and IL-13. So these cytokines, which we'll be calling Th2 cytokines, will also help provide co-stimulation and lead to B-cell class switching towards IgE. So I mentioned a few of these, but primarily these cytokines include IL-4, IL-5, IL-9, and IL-13. And without IL-33 and DAMPs, or the damage-associated molecular patterns, dendritic cells will typically actually induce regulatory T cells, not a Th2 response. So these responses can occur as a very local response through B cells, and often this occurs locally in tertiary lymphoid structures. So these are going to be germinal centers or lymphoid tissues that develop in sites of inflammation. Another common feature of many allergens is that they're proteases. So most airborne allergens, as I mentioned before, are soluble proteins on a dry particle. So this soluble antigen can very easily get into the body. And this low dose, so the body seeing this antigen very small amounts over time, leads to skewing towards the Th2 response. And protease antigens make this all more possible because they can disrupt tight junctions in the epithelium. So this allows them to pass more easily into the body where they can induce allergy. So here we're going to start out looking at some sensitization. So here we see an enzyme, in this case DIRP1, which will cleave some of the tight junctions, allowing it to enter the mucosa. So then, once in the mucosa, this antigen is taken up by dendritic cells and antigen-presenting cells, and then they are taken to the lymph nodes for antigen presentation and the production of a Th2 response. So then these Th2 cells will then induce B-cell class switching towards IgE. Finally, these B cells will then mature, and IgE-producing plasma cells will actually travel back to the mucosa. And these cells will then produce this DIRP1 specific IgE antibody, which will bind to the Fc epsilon receptors on the surface of mast cells and basophils. And this primes the tissue so that next time the antigen, in this case DIRP1, enters the mucosa, these cells will be poised to bind to that antigen and immediately release granules, which causes symptoms in an allergic response. So here, this gives you guys a good overview of sensitization. So, as I mentioned before, on the first exposure to an antigen, there's no symptomatic allergic response. Instead, you need the development of an adaptive immune response, especially an IgE response, for that to occur. Okay, so which factors then contribute to the development of allergy? So not everyone that encounters an antigen will become allergic to it. So as you might expect, there are genetic and environmental factors that contribute to susceptibility to allergy. And there's one example of a genetic disease called Netherton Syndrome, which is a mutation in SPINK5. And this protein encodes a protease inhibitor in skin. So if you guys remember back to the last figure I just showed, the antigen we were talking about in that case was a protease that was able to disrupt the epithelium in the skin or mucosa. So in this disease, we have protease inhibitors that are no longer functional. So we lack the ability to block those proteases from entering. So this then leads to keratinocyte shedding and disrupted barrier function. And patients typically develop multiple allergies, especially allergens that they come into contact through the skin. So here's an example of a patient with Netherton Syndrome. And so you can see the very obvious rash and keratinocyte shedding that's occurring in this patient. You can also see here on the bottom this histology section, which shows the thickening of the epidermis. And you can see at the top as well, such as over here, how we see some keratinocytes that are starting to shed more than normal off the top layer. So this disrupted barrier function really leaves these patients vulnerable to the development of allergy, as more potential allergic antigens are able to get into the body. So there are some other genetic factors that contribute to IgE-mediated allergy. For example, some alleles at IL-33 receptor and IL-13 loci are associated with allergic asthma. And there are also ethnic differences in susceptibility genes. So associated genes typically are actually cytokines that are important for switching to IgE. So these are going to be the cytokines we talked about earlier, your IL-4, IL-5, as well as some subunits, such as P40, which is a subunit of both IL-12 and IL-23. Okay, so as we mentioned with the autoimmunity and immunodeficiency lecture, the HLA variants are very important for differences in the immune response. So in this case, HLA-2 variants are associated with responses to specific allergens. And this is likely due to peptide presentation. So different HLA variants are able to bind to different peptides. And so it's possible that some variants are able to present more peptides from allergens and therefore promote a Th2 type response. So there are also environmental factors that interact with genetic susceptibility. So here's just a figure showing an overview of this. So we have our genetic susceptibility, which can lead to atopy, but it's also synergistic with things in the environment. So they have an example here, different infections that together can lead to more development of allergy. And it's thought that environmental and genetic factors each contribute about 50% of the risk of development of atopy. And the prevalence of allergic disease has actually been increasing steadily for the past 50 to 60 years. And there are several hypotheses as to why this is, but the most common or most well-known hypothesis is called the hygiene hypothesis. And the hygiene hypothesis states that now that many countries are more developed, we have better sanitation, this leads to less exposure to microorganisms, which therefore leads to skewing of the immune response towards a Th2 response, which causes allergy. And to support this, we actually see that children that grow up in areas maybe that are less developed or maybe grow up in an agricultural area or on a farm, they're exposed to more pathogens and typically have less allergy development. So some of these microorganisms that might be important are, in particular, your intestinal parasites. And these are really decreased in the last 50 to 60 years with the improvement of sanitation, and it seems to be that this correlates with an increase in allergy. So along these lines, there are several other environmental factors that can contribute to allergy. So I mentioned these microorganisms. Well, actually, repeated exposures to antibiotics can increase the risk of developing asthma. So in patients who receive antibiotics repeatedly as they're growing up, it's thought that this disrupts the microbiome and therefore can predispose the child towards allergy. A few other factors that can contribute include diet, exposure to different allergens, the pollution in the atmosphere, as well as exposure to things like tobacco smoke. And these environmental factors really synergize with genetic factors, including some null mutations in GSTP1 and GSTM1, which prevent oxidative stress. So now we're going to shift towards discussing a bit more about IgE-mediated allergic disease. So, IgE engages unique effector mechanisms when it binds to the Fc receptor. So, as I mentioned before, IgE binds to the very high affinity Fc epsilon receptor on cell surface. So most antibodies that we've talked about throughout the course engage Fc receptors after binding to their antigens. But IgE is different. It actually binds to the Fc receptors first. It's then the cross-linking of these receptors by an antigen that leads to mast cell and basophil activation and degranulation. So, as I mentioned before, this typically occurs at sites of antigen entry into tissues. And for example, this is really prevalent in the mucosa. Mast cell degranulation then proceeds to recruit other cells important for the allergic response, including eosinophils, basophils, and Th2 cells. And high IgE levels, like seen in an atopic individual, can then lead to higher expression of Fc receptors. So I've mentioned a few different cell types so far as we've been going through this overview. Now we're going to take a step back and talk more specifically about how these cell types are involved in allergy. So we're going to start by discussing regulatory T cells. So Tregs are very important for controlling allergic responses, and they actually might help prevent mast cell degranulation. So we see in atopic individuals that the circulating leukocytes actually tend to skew towards a Th2 response. And these atopic individuals might actually have a defective regulatory T cell in which these Tregs are unable to suppress the development of a Th2 response. And we also see that mice that are a knockout for FOXP3, which means that they lack regulatory T cells, have spontaneous allergic inflammation that develops. And it looks like, mostly through mouse research, that therapies to enhance regulatory T cells may actually be beneficial for asthma and allergy. Okay, so mast cells actually are developed from the hematopoietic stem cell niche, but they mature locally in tissues. They're tissue resident. They also reside near barrier tissues such as the mucosa or around blood vessels. And it seems that these mast cell precursors actually can reside in the tissue before differentiating and they will very rapidly differentiate into full mast cells upon an allergic response. And degranulation of mast cells actually occurs within seconds of an antigen IgE cross-linking. And this is particularly fast because that IgE is already bound to the surface of mast cells. And I've mentioned these granules a few times. We see that they can contain histamine, serine esterases, and different proteases. So, what are the effects of degranulation? So, histamine is the primary chemical in these granules that causes symptoms of an allergic response. And histamine acts on 4 different receptors found in different areas. These are called H1 through H4. So we see that these receptors, when they're located on blood vessels, will increase blood flow and vessel permeability. The histamine receptor engagement can also increase antigen presentation by dendritic cells and promotes priming to Th1. We also have receptors for histamine on local neurons. So this is what leads to the stereotypic itching and sneezing response. There are also some receptors in the skin leading to atopic dermatitis and urticaria, which, as I mentioned before, is hives. And sometimes this can have some factors of autoimmunity as well. So proteases in these granules can then activate other enzymes such as metal metalloproteases, which break down the extracellular matrix. And interestingly, these proteases can also break down venoms. So one stereotypic allergic antigen is a bee sting or bee venom. So these proteases actually help break down that venom. And these granules also can release cytokines as well as lipid mediators like prostaglandins, which initiate an inflammatory response. And this causes things like smooth muscle contraction, especially of the airways, and increased permeability of the blood vessels. So these responses are nicely summarized in this figure. So here at the top, just by mast cell degranulation, we have multiple different effects that can happen. So in the gastrointestinal tract, we'll see responses such as increased fluid secretion and increased peristalsis. So that's the contracting and relaxing of the gut. We'll talk about that a bit more when we discuss food allergy. We also see in mucosa, such as nasal passages, that these airways contract. So the airway diameter is smaller and there's more mucus secretion. And in blood vessels, as I mentioned, we have increased blood flow and increased permeability.